Hello viewers, welcome to Biodrunks. Let's learn some aspects of biotechnology. For your reference, I'll be providing notes, PDF link in the description. This video is also available in Hindi and Telugu. For more videos and continuous learning, please like, share and subscribe. Let's start!
In the previous video, we were learning about cDNA library synthesis. In today's video, we will be learning about genomic DNA library. So what is this genomic DNA? Genomic DNA means the whole DNA of an organism, or I should say total DNA, okay, of an organism. Now, we are constructing a library from the total DNA of an organism. Okay, so how do we do this and what are the process steps? So first learn about genomic DNA library. Genomic DNA library means the total DNA of an organism. And how do we obtain it? Why do we use it? And what is the most important sequences we get from that? So as I told you, genomic is the total DNA. That means it has all of the constituents like expressed genes, non-expressed genes, and then exons, introns, and you have promoters. Okay, let me write it one by one. Promoters, terminator, regions, and then intervening DNA sequences. So you have almost all the important things that are required in an organism or organism genomic DNA. Okay, so why are we using it? So genomic DNA, they are used because they can find novel products such as antimicrobials. They are used to know about the biochemical pathways for the production of biofuels, and there are many functions as of now. Okay, let's learn one by one the steps. So when I talk about steps in genomic DNA construction, it's very easy. Okay, so what do? We first need to do is the isolation of the genomic DNA. Always remember, isolation is quite important. From where are you isolating, which organism or which tissue it is? That's very important. Once you isolate the genomic DNA, then you need to purify it, and then we have fragmentation. So in the previous videos, you can find a detailed explanation of how the DNA fragments are being cloned and how to work on that. Like what are the different methods in which we are fragmenting the DNA. That is, the video name is Generation of DNA Fragments for Cloning. Okay, but then also I'll be giving a brief about the fragmentation here so that you can link with the video. Now, this DNA has to be fragmented. After fragmentation, what you are doing? You have to get the proper size. Okay, so for that, we do gel electrophoresis to get a proper size. And then after this, we elute the fragment needed and then we clone into a suitable vector. Okay. So these are the basic steps about genomic DNA library construction. Let's learn about fragments. How do we get the fragments? Isolation and purification is the basic method, but fragmentation, it has many. It has two types. One is physical and one is chemical or enzymatic. Okay, so when you do physical, all these methods have been dealt. I'm just giving you a brief. When you do physical, you do mechanical shearing or you use sonication process, okay, or you use needle for that, needle syringe, needle gauge, okay, and we need to get an average fragment size of nearly 20 kilobase pairs, and this is the desired size for lambda vectors. Okay, when you do enzymatic, that means what are we using? We are using restriction enzyme. Now, what happens when you digest it totally with a restriction enzyme? For example, this is your genomic DNA. It has restriction sites. Okay, now imagine for a particular restriction enzyme, you have multiple restriction sites in a very small interval. So what happens when this DNA, when you cut it with that, you get smaller fragments. So these small fragments cannot be cloned into a vector because they don't represent the total sequence or the required sequence needed for cloning. So that's why we don't go for total digestion. We always do partial digestion of the DNA. Okay, partial digestion is quite important and we need to know more about it. Okay, so what does we do? Here we use like whenever you're doing it, here you use known quantity of restriction enzymes. Okay, when you use a known quantity of restriction enzyme, you obtain fragments of ideal size that is needed. Okay, now after the digestion, we do gel electrophoresis where we elute the product. Once you get it, if you don't have the proper like size or blunt ends or sticky ends, however you want it, you can convert them by using linkers and adapters. This video is also dealt before. Please check out the links for different videos in the playlist. So do start from the beginning so that you can understand each and every step here. Then now we obtain the proper fragments that are needed and then this is been cloned. Now for cloning of the genomic DNA, we use different types of vectors. Always remember you have different vectors for different sizes. Okay. Now this video about vectors will be dealt in very detailed in the upcoming videos. But here let me just give you a briefing about what are the vectors we are using. So you can use a lambda fake, but the size should be nearly 20 to 30 kilo base pairs. So we usually don't use lambda fake because it's of very less size. We start with cosmets, which is up to 40 kilo base pairs. Okay, and then these cosmets, and then you can use phosmets. Okay, which is 35 to 45 kilo base pairs. Bacterial artificial chromosome, which is up to 300 kilo base pairs. P1 artificial chromosome, which is up to 300 kilo base pairs, and then yeast artificial chromosome, which is up to 2000 kilo base pairs. So we usually these are used to handle longer DNA inserts. Okay, so much larger than lambda fake. Okay, so they require lower number of recombinants to be screened. Okay, now if you have a lower size, you have lot of recombinants that have to be screened, and we don't want it that way. That's the reason we usually go for larger size. Here you can use cosmets, bacterial artificial chromosome, P1 derived artificial chromosome, and yeast artificial chromosomes for the genomic DNA library construction. Now you get a particular clone. Okay, so the number of clones to be pooled depends on the size of the DNA or the genome. Okay, so we can find it. How? So whenever you want to find the number of clones required for library, that is F, it will be equal to the genome size, that is the total size of the genome, divided by the fragment size that you get after cloning. For cloning, whatever the fragment you're using, that size, which will give you the minimum number of clones required. So this F means minimum number of clones required. Okay, this is one formula. Okay, and you can also calculate the probability of any DNA sequence in a random library of an n-dependent recombinants. Okay, so this can also be calculated by n is equal to ln 1 minus p divided by ln 1 minus 1 by f. So here where n is your number of clones, okay, p is probability that the given gene will be present, okay, probability of given gene is present. Okay, so this was about few steps. Now, you have two types of libraries. One is amplified library and one is subgenomic library. So what happens is whenever you are cloning a particular DNA or particular fragment, what happens is it has a low titer and they are a bit unstable even. Okay, so the stability and titer they can be increased by amplification. Okay, so for this, for example, the fakes or bacterial colonies are plated out several times. Okay, and the resulting progenies are collected to form an amplified library. So the amplified library can be stored almost like for long shelf lives. It can be stored for long shelf life of fakes. Okay, now what's the next subgenomic library? Subgenomic library means it only represents the fraction of genome. It has only the fraction of genome. Okay, so subgenomic libraries can be achieved by multiple sequential digestion when the information of the restriction map of the sequence of gene of interest is known. So here you know the sequence of the gene of interest and therefore according to that you use restriction enzymes and you do digestion. Okay, so till here this was all about genomic library construction and what are the types and why everything. Now what is the application of genomic DNA library? So here the applications are very simple. You have the complete genomic sequence of an organism. You can determine it. You can study transgenic animals through genetic engineering. Okay. And then you can know the functions of the regulatory sequences. You can know genetic mutations of a particular like organism or the thing. Okay. So these all are the applications of the genomic DNA library. Now, how it differs from cDNA library? See, whenever you talk about cDNA library means it consists only of the structural genes. Whereas genomic DNA libraries, it consists of all genomic sequences. Now, you have is this size is smaller when you compare it with the genomic DNA library. So big. Here you don't have your introns. Introns are absent. Here you have, yes, you have the introns here. Okay. So this sequence is present in one or more clones and only in one clone. So here only in one clone and here you have one or more clones. Now, what about, see, here you get it from mRNA. There's a lot of it. But here you get it amplified by the genomic DNA because it has the whole genome. Okay. I think almost we dealt with almost all the points. Here you use reverse transcriptase. Here you don't use reverse transcriptase for the process. Okay, this is very easy. Now here, because you have smaller fragments, so you use vectors that can obtain or that can handle smaller fragments. Because here the fragments are big, you need like YAC, VAC, okay, cosmics. Yeah, here you need lambda phage, or you can even go for phagmits and plasmids because they can obtain or harbor smaller fragments. Okay, so this was all about the differences. Now, hope you understood every aspect in the genomic DNA library construction.
This was all about today's video. Hope you liked it. For more such interesting video, please like, share and subscribe to BioDrunk. Let's meet in the next video. Bye-bye.